Okay, we started. Good evening, everybody. Thank you for coming to the curtain egg of the Bayliss Youth Lecture. My name is Dino Spagnoli, and I'm the chair of the Chemistry Education Group of the WA branch of RACI. I'd like to acknowledge that we are meeting on Noongar land, and the Noongar people remain the spiritual and cultural custodians of their land and continue to practice their values, language, belief, and knowledge. Before I forget, I'd like to thank big thanks to Adjunct and Curtin University for supporting this event, and through their generosity we're able to offer guests a light refreshments after this lecture, and hopefully we can continue the discussion about the fascinating talk that we're going to see this evening. It's my great pleasure to introduce Professor Charlie Bond and Associate Professor Archer Fox, who will give an excellent talk, and I know this because I've seen it before, on a very topical subject. So please join me in welcoming them to the floor.
Thank you. Thanks, Dino. Okay, here we go. So welcome, everybody. We'd also like to acknowledge the Noongar people as the traditional owners of the land. Malak Jerabin Wajuk Buja Ninin. We really enjoy living on Noongar land, and we get to work in this beautiful place, and we get to work in the areas of chemistry, biochemistry, and cell biology. So my background is as a chemist originally. I did a degree in chemistry, but at the end of that, as I started a PhD, I became a bit obsessed in how biology comes out of chemistry, and I decided that all the interesting chemistry questions were in biology. And so I've moved ahead from there, and I've spent my career applying chemistry in biology. Archer has started off doing molecular genetics and then molecular cell biology. And she discovered that lots of the answers to the questions she was addressing had aspects of chemistry in them. And luckily, we got together and got married. And so we've been able to work in this area together for many years. Won't say how long. This is a great opportunity for us to talk about really the significance of chemistry and an important subject to humans, which is how on earth did we get to the point where suddenly when a novel virus hit us and we had a pandemic, how did we quite so rapidly get to the point that we were able to come up with a vaccine to treat it? So, Archer's going to start with some of the background. We're going to tag team throughout this talk just to keep you on your toes. So,
yeah. Okay. So I'm going to start us off. I want you all to cast your minds back to the beginning of 2020 when this new virus emerged, and we were all quite worried about would we even be able to make a vaccine. We'd never had a vaccine against a coronavirus before, and the quickest any previous vaccine had been developed was within a 4-year time period. So I don't know, I remember, I guess you guys should remember that feeling of let's put our faith in science, let's hope there's a vaccine that can be discovered, but it was never a guaranteed thing. Roll on to the end of 2020, and we had spectacular results from phase 3 clinical trials for the first COVID vaccines. And we then shifted to a mood of skepticism, where many people were wondering if this vaccine had been rushed over the course of 2020. And people were thinking, I'll just wait to see, I'll let other people get it first. People were commenting that, we've had so much research on HIV, but we still don't have a vaccine against HIV. How can we have a coronavirus vaccine made so quickly? And really what our lecture today is going to show you is that there was decades of scientific research in many key areas that all came together beautifully to actually help this effort of developing the coronavirus vaccine. So we are focusing in this lecture on the new mRNA vaccines, and that's partly because that's mostly what we have received is the mRNA vaccines. And we'll try and explain a little bit about why they are also quite exciting for future therapeutics and vaccines as well. So some of you might know that the mRNA vaccines that you've received, this is the Moderna and the Pfizer-BioNTech vaccines against COVID, were actually designed over the course of a weekend. So incredibly quick to design. The first batches of those vaccines were made in the next few weeks. They were tested in animals and then tested in humans throughout 2020. So it was a very quick, very rapid process. We're going to explain to you everything that led up to that weekend where they were designed and all the science and discoveries that enabled the design to happen over the course of the weekend. And there are really three main players, three heroes to our story. So we have proteins, in particular the spike protein of SARS-CoV-2, which you can see here. We have RNA. This is part of the sequence of the Pfizer-BioNTech mRNA vaccine. And we have lipid nanoparticles, or lipids. So these are three chemical entities we're going to be discussing: proteins, RNA, and lipids. And we're going to go through our talk explaining the science behind them and how we got to where we did. There's one more fourth hero to the story, which I want you all to guess as we go along what that might be, and we'll reveal it at the end, what the fourth ingredient is that has led to the success of these vaccines. So before we get into that, I want you to think about vaccines. This is our first ever vaccine developed for humans, and does anybody know who this man is here who's pictured injecting people with cow elements?
Jenner.
Yep, Edward Jenner. So he discovered that if you take the serum from cows that have been infected with cowpox and inject it into humans, you can protect them against smallpox. But obviously, if you can see from this cartoon at the time, there was a lot of concern about this. You can see these cows growing out of the sites of injection here. We as humans have had healthy skepticism of science and innovation and new discoveries, and you can see that that continues with vaccine hesitancy today. But roll on from here, we have actually an amazing array of technologies now to make modern vaccines. But still we cannot make vaccines against some viruses. So HIV is going to be a little bit of a theme in our talk, and I'll be explaining a bit about HIV. I also want to draw your attention to this timeline down the bottom here, which shows you that several of the viruses that we're dealing with at the moment have only emerged very recently. So Ebola in 1976, HIV in 1982, bird flu H1N1 in 1997, the first SARS coronavirus in 2002, MERS, the Middle East Respiratory Syndrome coronavirus in 2012, and then of course SARS-CoV-2. What is interesting is that we actually have vaccines against all of these modern viruses except for HIV, as I said. Some of the vaccines are not made because they're not needed. So SARS was eradicated. There is no need to make a SARS vaccine because that virus no longer is a problem to humans. And MERS is a really, really nasty virus with 35% fatality, but has a very, very, very rare occurrence. So those vaccines are not made. But we do make now Ebola, H1N1, and of course SARS-CoV-2 vaccines. So the reason I put this up is also to explain how research into viruses more broadly and in how to make vaccines more broadly has helped us to make the SARS-CoV-2 vaccine, and in particular, as I stressed, HIV. The billions of dollars that have gone into HIV vaccine research, even though they have not yet given us an HIV vaccine, were instrumental in helping us get a SARS-CoV-2 vaccine. So we have this array of technologies now for making vaccines. We can grow viruses in the lab and then inactivate them. We can grow viruses and fragment them. We can make what we call the genetic viruses, of which the mRNA is an example, or the DNA for the AstraZeneca. We can make recombinant proteins, which is depicted down here. So there's all these different methods, but the one thing that they have in common for any vaccine is that it has to ultimately end up in production of a protein, because our immune system will recognize a protein antigen and develop immune memory of that. So whether we have a piece of DNA that instructs our cells to make protein, or a piece of RNA that instructs our cells to make protein, or a virus with proteins on the outside, or some protein that we made in bacteria and purified and injected, it actually all comes down to protein. So that is why I'm going to hand it back over to Charlie, and he is going to give you a talk about proteins, and then we'll get on to the RNA part of the story.
Great, thank you. So I'm absolutely obsessed about proteins. I think they're amazing, and I could bore you for hours. I'll try not to. Hopefully I'll not bore you for 10 minutes and you'll be equally enthused. So proteins do nearly all of the interesting jobs in your cells. They do all the communication, they're the infrastructure, they're the buildings, they're the roads, they're the machines, they transmit energy. They're an amazingly diverse array of molecules, and you can see a picture of a bacterial cell. This is an artist who's a structural biologist who does art, and he's drawn a picture of a bacterial cell, and lots of the stuff you can see is all these proteins jammed in together, making the cell work. So proteins arise because of the code that we bear in our genome. So our DNA, as we all know, has a code, but what's that code for? Well, to one extent, it's for coding proteins, so it gives the instructions to build these proteins. So we'll talk about this idea of the central dogma of molecular biology a bit later, but it basically tells us the relationship between the sequence of our DNA in our genome and how that ends up turning into the sequence of some proteins. And so the sort of simple analogy for that is the DNA is like the recipe in the recipe book, and the proteins are a bit like the meal on the table. So I showed you the DNA sequence and that there's a kind of a protein sequence, and that's because proteins are polymers. So they're polymers of amino acids, and amino acids themselves are called amino acids because they have an amino group and an acid group, and they condense together in this condensation reaction, called condensation because water is evolved, forming an amide bond. And so now you've got a new, we call it a peptide, and it's got an amino group and it's got an acid, and so more amino acids can join onto it, and you can get an infinitely long polymer in theory. The bits I've drawn in colour on here we call side chains, and those are bits that are chemically different between the different types of amino acid. So you might have heard of the amino acid tryptophan. That's got one kind of side chain, and the amino acid glutamic acid has got a different kind of side chain. So if we join them all up in a polymer, we can end up with a sequence like this, where we have different chemistries shown here just by different coloured blobs in the polymer. So you can think of it as something like a charm bracelet with different charms. And the key thing, the properties of this protein are dictated by the sequence of the colours, and we call it the protein sequence. And as I mentioned before, that's derived from the DNA sequence. So we have 20 amino acids. Basically 20 different types of amino acids account for most of the amino acids in proteins, nearly all of them. So if you have a protein that's 10 amino acids long, how many possible protein sequences are there for that? Anyone good at permutations? Maths methods?
A bunch.
That's a very good answer. A significant bunch, right? It's 20 to the power 10. So that's 10 million million. So there are 10 million million possible proteins that size. Now in humans, we only have 20,000 different proteins, and they're a lot bigger than that. So So nature has kind of selected for a very small repertoire of particularly useful proteins that it can use. But if you think like a combinatorial chemist, this is an amazing resource for building chemistry, and that's why nature has adopted it. So what's really cool about proteins is they're not just some polymer like polythene or PVC or something. They are these perfectly, beautifully structured molecules. So depending on the sequence of those amino acids, they will form up into a unique three-dimensional structure. So here's an example of a 46 amino acid protein. And we use a one-letter code because there are only 20 amino acids. We can use a letter each, and that tells us the sequence of the protein. And this is what its structure looks like. And there's a couple of different representations here. This is a structure showing all of the non-hydrogen atoms. But we can show more simplified structures which show that it's actually a polymer. It's one single chain, long chain, that just folds up. And if I were to synthesize chemically this sequence of amino acids and put it in a buffer in a test tube, it would fold up into this conformation. So years ago, my daughter, when she was little, had these kits for making a necklace where you put all the beads together. And I remember thinking about these different coloured beads and thinking about proteins and thinking it wouldn't be cool if you had a toy where, depending on the order of the coloured beads that you put together, you then just threw it on the floor and it automatically turned into a dog or a house or a car or some other shape. That's what proteins are. Just 20 building blocks, and they can form the most amazing complex structures repeatedly. So how do we know this about the structure? Like, proteins are tiny, right? They're at the angstrom nanometer scale. And so we have a whole bunch of methods that we use to analyze them. There's a method called crystallography. There's a method called cryo-electron microscopy. And we can do crystallography in our labs here in WA, but most of the time we actually have to use the synchrotron in Melbourne, the Australian synchrotron, which is this huge particle accelerator the size of a football pitch, throws out really intense X-rays, and we can use those X-rays to analyze our crystalline protein samples. And then we have cryo-electron microscopy, which is a newly introduced method, and we've got a brand new facility at UWA that can be used. So thousands of labs around the world use these methods to understand the structure of proteins. And when they do that, and they want to publish their results, they have to deposit those structures in a public database that's available. You could go and if you Google the protein data bank right now, you'll find it, and you'll be able to get access to the structures of any protein that anybody has determined the structure of, particularly if they want it published. And so you end up with tiny little proteins like this one that I showed you before that's 46 amino acids. Here's one we worked on in our lab, which is 620 amino acids, so that's about 20 nanometers in size. Here's some other big molecular machine that helps fold proteins up, and that's 25 nanometers in size, and it's about 8,000 amino acids. So 8,000 amino acids, but only 20 different amino acids, and the magic is in the order in which they're joined together. So very rapidly, once COVID had been recognized even, people jumped on the job of understanding the structure of the proteins in the virus. And so as soon as the genome of the virus had been published, people already knew something about viral proteins. They were able to go in and start working on them. And so this is a schematic of all of the proteins in the different parts of the genome of the virus. And there are two kinds of proteins. There are the structural proteins, which are they do what it says. They're the structure. They make the shape of the protein and hold it together. Of the virus and hold it together. And then there are the non-structural proteins, which are the ones which do the chemical jobs, like chopping up other proteins and transcribing RNA and all sorts of other things. So this schema shows what we know about the structure of these proteins as of about a year ago, actually, possibly even longer. There were experimentally determined structures of the SARS-CoV-2 proteins, or the SARS virus, the original SARS virus proteins, which are very similar, or in some cases, very good predictions of the structures. But this great resource and the sharing of data is one of the things that helps accelerate science when something like a pandemic strikes. So here's an animation of the virus. This was done by Brady Johnson, who's a PhD student in my lab at UWA. He really likes trying to work out how to visualize these proteins. And what we can see here is a coat of lipids around most of the virus, and sticking out from it are these spike proteins. And so the spike proteins have been one of the keys to vaccine development for different diseases for a long time, because in viruses they stick out. They're the spikes that stick out. They're the bit that the body sees first. They're the bit it can recognize to detect a virus. That's why they're important for vaccine development. So a bit of history now. Anyone know who any of these guys are?
Tony Fauci, yeah.
Yeah, that's the one that people don't normally see because his hair's changed colour. This one, Bill Clinton. So this was back when he was president and Al Gore was his vice president. I'm not sure who that is. But this is Anthony Fauci, who you'll have seen on the TV a number of times over the last couple of years, leading the SARS-CoV-2 battle in America. But this was back in the 90s, where Fauci was working at the NIH in HIV research, and at some point, Bill Clinton got in touch with him and said, look, we've known about this virus, HIV, for a decade now. Why haven't we cured it? What will it need to cure it? And over the course of a few years, they cooked up a plan to set up a major facility called the Vaccine Research Center, which is hundreds of millions of dollars, sort of immediately built and employed dozens of expert scientists, and their job was find a cure for AIDS. And that massive investment is one of the things that's helped us when the recent pandemic started. So there are a couple of guys here, Barney Graham and Jason McClellan at the Vaccine Research Center, were working on the HIV spike protein with a view to making a vaccine for HIV. And if the idea was, if you make the spike protein on its own, then you can use that to develop a vaccine that will train your immune system to recognize HIV in the cell. But they never had any success. They had lots of struggles. They basically couldn't do anything with HIV. They managed to change tack and think, all right, we'll try a different virus. And there's one called respiratory syncytial virus that you probably never heard of. It's a related virus, it's very rare, and it's a childhood disease. They were able to understand something about the spike protein from RSV. And they did some really important work that has had a more resounding effect recently. So they realized that this spike protein is actually very flexible. So I showed you some structures of proteins before, and they're kind of a rigid structure. But actually, proteins are flexible. They can kind of breathe, and some of them are really flexible and actually do a kind of shape-shifting thing. And the spike protein is an example of that. So it turns out that they could show that if you just make the spike protein for RSV, it folds up in this particular shape here that they called the pre-fusion shape. And if you use that to develop antibodies, the antibodies recognize these regions or epitopes that are colored here. However, when the protein is actually interacting with your cells and with your immune system, it goes into this fusion conformation called or post-fusion conformation. And that's the same protein, but it's just shifted into a different conformation, like me going from this to this. I'm still me, but I'm in a different conformation. When that happens, the number of epitopes massively reduces. So if you use this as the basis to train your immune system, but this is what you're trying to recognize, doesn't work so well. So they had to think about ways to trick the protein into adopting the wrong conformation, this conformation, so that it could be used to train the immune system. So, how do you stabilize a floppy protein? Molecular handcuffs. It's a kind of lame analogy, but in a way it's accurate. At least one of the techniques they used, molecular handcuffs, is accurate. So the first technique I'll talk about is if we drill into the 20 amino acids that I talked about before, one of them is special, proline is special. And here's a short bit of protein. These are all amino acids joined together by amide bonds. Amide bond there, there's an amide bond there. And here are the side chains coloured. The proline side chain sticks off, and then it comes around in a circle, and it joins up again with the nitrogen. And that means the protein at this particular point is less flexible, right? It could normally spin around like this, but because it's joined together, it no longer can. So if you introduce proline amino acids into a protein, then you can stabilize its structure. And that's exactly what they did in a study on MERS spike protein. So they were able to stabilize it by introducing 2 consecutive proline residues. And that just stuck it in the right conformation for vaccine manufacture. So moving up to now, the protein that's being used basically for the SARS-CoV-2, or one of the proteins, is called hexapro. And they used the same trick applied to the SARS-CoV-2 spike protein, along with some other tricks. So there are a number of prolines that have been introduced. So a particular amino acid in this has been changed to a proline. And that makes the protein more rigid. There's another type called the ionic zipper, where there's a positively charged amino acid in one part of the protein, and nearby there was a neutral amino acid, but they change it to a negatively charged amino acid, and that causes a charge pair which can interact ionically and stabilize the protein. The one that's more like handcuffs relies on another one of the 20 amino acids called cysteine that has particular properties. Cysteine has a thiol group in the side chain, this sulfur, and that means it can be oxidized, and it forms a covalent bond between the 2 sulfurs, which is called a disulfide linkage. So that covalent bond can permanently link 2 parts of the structure together, of the protein together, irreversibly. And the other one's a bit more subtle, can be described as molecular polyfiller. That's where you know the shape of the protein, and you know that there's, for instance, a sort of a valley in the protein here, and you can see that there's space that could be filled by some atoms. And so if you change one amino acid, like an alanine here, to a phenylalanine, you can fill that space, and that can cause a stronger interaction between this part of the protein and this part of the protein. So all of these techniques were used to make hexapro, and that's a successfully stabilized protein. So that's great. We've solved all the problems. We can make a protein that has these properties. One of the problems we have is proteins are actually expensive to produce. So many of the proteins that are required for these purposes need to be expressed in mammalian cell culture. It's extremely expensive. So, for example, Herceptin is a protein drug that's produced for the treatment of breast cancer. And an annual dose of Herceptin is $80,000. So making proteins as therapeutics is a very expensive approach, even if it's sort of got some great upsides. So one of the key innovations that happened that's got us where we are with mRNA vaccines was people thinking, well, hang on, proteins are coded by DNA, and an intermediate to that is RNA. So maybe we, instead of using cultured cells, we can trick our own cells to be the drug factory. If we can put that coding sequence into our cells will make the protein, and we don't have to spend $80,000 on mammalian cell culture. So the central dogma of molecular biology that I mentioned before is the explanation of how DNA codes for proteins. And this is where I'll pass back to Archer to describe a bit more about that.
Okay, so now we're going to shift our attention to RNA. And I'm talking about RNA because I am an RNA biologist, and I spent most of my career working on RNA. I actually discovered a structure that we have in our cell nuclei, which is built on a particular RNA. That RNA doesn't code for protein. It's a very special kind of RNA. It just stays in the nucleus where it was born. That's not the topic of today's lecture. So I know you're probably all familiar with this, but I'd like to go through it again, what Charlie's already referred to as the central dogma of molecular biology. So this is just really describing how the instructions in our DNA are read out into this intermediary messenger RNA, and then that is the instruction for making our protein polymer down the bottom. And you can see that the RNA is very similar to the DNA, except it's single-stranded, and instead of having these T's, we instead have U's. And I'll talk a little bit more about that in a moment. So chemically, RNA is actually very similar to DNA, incredibly similar. Just like proteins, DNA and RNA exists as a polymer. We call it a polynucleotide, and you can see it here on the right of the screen. And you can see there's this backbone, and then you can see these side chains sticking off here. So very similar to proteins, but chemically very different. Now, if we drill into one of these nucleotides, as we call it, and we just shift to the left here, we have this pentose sugar group, and then we have a phosphate off here and a base, what we call a base. So it's the base plus the, it says the base, which is the CTU or AG code. And if we look down here, we have the only difference between DNA and RNA. So DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. And it's just this little hydroxyl group here at this 2' position that is the only difference. So it's a very, very subtle chemical difference, but it actually has a very dramatic effect on this polynucleotide chain. Because what it means is that water can attack this, this chain much more easily. So RNA is much more easily degraded than DNA. We don't want our DNA to be degraded. It has the instructions that tell our cells what to do, and it has to be passed on to the next cell when it divides. But RNA is okay for it to be degraded because it has a transient and temporary job. It makes an instruction to make a protein, but then it's no longer needed after its job is done. So this is a beautiful animation that was produced from the Walter and Eliza Hall Institute, and it shows the process of gene transcription, which is RNA being made. So this is a little enzyme called RNA polymerase, and it's running along this DNA strand, and it's creating this new RNA strand using the DNA as a template. And in a minute, we'll zoom in here, we'll open up the enzyme. You can see here this complementarity. We've got the DNA template, and here are these little RNA nucleotides, each being made, added on, complementary to the DNA template. Now, this is that production in real time. This is what's happening in our cells all the time. We are making thousands of these RNA molecules using this enzyme. And as scientists over the years discovered this process, actually quite early on, they started to think, well, why can't we make these RNA molecules in the lab? And we could purify these enzymes. And so even from the 1970s, scientists were putting into a test tube a bit of DNA and an enzyme and watching the RNA that was made. We call it in vitro transcription, or transcribing RNA in a test tube. So we could do this for 30, 40 years, but we didn't have the ability to take that messenger RNA and put it into our cells and instruct our cells to make proteins. And this was the reason why. Because scientists quickly discovered that if they took some synthetic lab-made mRNA and put it into our cells, whether it's animal models or humans, that our cells have inbuilt defense mechanisms to try and attack foreign RNA. Of course, this is what our cells do when they detect viruses. Virus RNA goes in, our cells immediately recognize that as foreign, start to build an immune response against it. And that's what was happening to the synthetic lab-made mRNA of those first experiments. We had a whole variety of different molecules that recognize these as foreign, and they start to initiate a process of cell death, and we get inflammation. But it is a bit of a mystery why this doesn't happen with our own RNA that our cells naturally make. Okay? This happens to virally injected RNA or synthetic lab-made RNA. So there was decades of work by RNA biologists like me trying to figure out why our own mRNA that our cells make doesn't trigger this response and all of our cells just don't implode. And they've figured it out. So it turns out that our mRNA that our cells make have little chemical modifications in them. And those chemical modifications are one way that our cells know these RNAs are good and natural, don't trigger the immune response. And these two people, Drew Weissman and Caitlin Carrico, figured out that they could fool our cells by making modifications in the lab-produced RNA. So instead of just making RNA with A, C, G, and U, they could make this slight modification, and I'll tell you about that in the next slide. And they discovered this back in 2005. So by incorporating a modified nucleotide called pseudouridine into in vitro transcribed RNA, we no longer got this cell death and inflammation. So here's a little bit more on the chemistry of those modifications. So I've got here the natural nucleotides, cytosine, adenosine, uridine. And then down here you can see some of the modifications. So quite subtle. 5-methylcytosine, you can see it has this methyl group here. N1-methyladenosine has a methyl group here. Or N6-methyladenosine up here. And here's the one that made the big difference in the synthetic RNA. It's pseudouridine. So in this case, it's just a different configuration of these atoms to make pseudouridine as opposed to uridine. So that was discovered in 2005. And then very recently in 2017, actually N1-methyl-pseudouridine was found to be the most effective modification for fooling our cells into thinking that the RNA we were putting into it was natural. So this breakthrough was absolutely pivotal for the ability for us to have mRNA vaccines. If we don't put in these modifications, then our cells will trigger an unwanted response. And this whole field of what we call epitranscriptomics, or understanding why we make these modified nucleotides, is a very vibrant field, and there are many more questions than answers about why we have all these different chemically modified nucleotides in our RNAs and what they're doing to our RNAs. And so, I encourage you to consider going into RNA chemistry because there's so much we still need to learn. So at the same time that we were working out about these modified RNAs, we actually were experiencing a biotechnology boom. And it was centered around these two companies, Moderna and BioNTech. So Moderna developed the mRNA vaccine. Some of you, hands up if you've had the Moderna vaccine. A few of you. And BioNTech partnered with Pfizer to make the Pfizer vaccine. So these companies were established back in 2008 and 2010. And both companies were established on this mRNA platform. And remember that Caitlin Carricker and Drew Weissman made their discovery in 2005. So it was with that discovery that then enabled these companies to say, okay, we now know we can make RNA in a lab, we can modify it, let's see if we can fix diseases with this. So these companies formed to focus on cancer. So they started to make mRNAs to try and cure cancer. But interestingly, when they started to test these, they noticed that they triggered an immune response in the whole animal, and they thought, okay, actually maybe this might be better for vaccines than cancer therapeutics. And they developed amazing pipelines over the course of this decade to design RNA, to test it in animals. They developed partnerships, like BioNTech developed a partnership with Pfizer in 2018, actually to develop a flu vaccine. Huge amounts of money went into these companies, and by the end of 2019, Moderna, for example, had $1.5 billion been invested in that company, but not a single drug had been produced from the company. So there would have been a huge amount of investment, but actually not a lot to show for it, just before the pandemic hit. But what these companies were doing is they were developing these pipelines to very rapidly design mRNA drugs. And they spent a lot of time investing in bioinformatics, so very sophisticated computer programs to design different RNAs. And they worked out the best elements to add onto different RNA sequences. So I haven't really gone into this, but here's a typical messenger RNA, and this central part is what we call the coding region, so that's the part that is the instruction to make the protein. And then at the very start, we have what's called a 5' cap, which stabilizes the RNA. Then we have a 5' untranslated region, which regulates the stability again, and a 3' untranslated region, and a poly-A tail. And all of these elements occur in RNA, but if you're going to make one in the lab, you want the best possible elements. And so these companies put so much effort into having these design platforms developed. And the other feature of these platforms is that they had to be rapid. Because remember I said they were formed to help with cancer. And the pipeline for cancer therapeutics that they were going with is that a patient would present with their tumour, that would get profiled, an RNA would need to be designed and very quickly made so that that patient could then receive the benefit of that treatment. So speed of design was built into the pipeline, which again comes back to the design of the vaccines being so quick. So that is it for the little RNA section. I'm going to hand back to Charlie. We're going to have the third element, which is the lipid nanoparticles, and then we're going to bring it all together at the end.
Okay, so lipids. Lipids turn out to be really important in this story because we know now that RNA is unstable, doesn't get into the cell very easily. And so we need to find ways to stop it being degraded and to persuade the cells to take it up. And nanotechnology turned out to be the field that could answer this. So nanotechnology is the study of things in the order of 1 to 100 nanometers. Now, proteins you could consider to be nanomachines or nanomolecules. I kind of think they are. They're self-assembling nanomachines that do really cool jobs. But it turns out that other kinds of molecule can also do this. And lipids self-assemble. That's one of the reasons nature uses lipids is because of the way they self-assemble, and that can be used for bio-nanotechnology. So lipids are these molecules that have non-polar tails and polar heads, and because the non-polar parts want to hide away from water, basically they self-assemble spontaneously when they're in an aqueous environment into different structures. So you can end up with things like these bilayers, which are large sheets. That's what binds around our membranes. All of our cell membranes have this lipid bilayer, where with the non-polar parts hidden away from the water. And you can make micelles from certain kinds of lipids, which have a similar thing, but they form a sphere, or you can have these liposomes, which are a bit like a bilayer bent round into a sphere. So they're all different kinds of shapes of lipid self-assembly. Just looking at this reminds me, two bears fall in the water, a white one and a brown one. Which dissolves first? The white one, because it's polar. Right. Sorry. Sorry, but I bet you remember. So here's another animation featuring lipids. So this is the virus again, an animation that Brady did, but showing the lipid the lipid coat on the virus in brown there, kind of undulating. So lipids can be very large, flexible objects. So academics have been working on lipids for a long time because they're interesting, and there have actually been some medical applications because relatively recently people discovered that a certain kind of very short RNA molecule in the order of 20 nucleotides could have very important medical effects. So it turns out if you can get specific short interfering RNA molecules into cells, they can change which genes are regulated. And that could be important for certain diseases like cancers. So people were already interested in delivering short RNAs. And Peter Cullis, a Canadian scientist, was involved in this research in using novel lipids as the technology to deliver siRNA. And he described relatively recently what was a fringe field back in the 1980s has turned into something that is mainstream now, because all of us have benefited from this work. So we've put the dates of when some things happened on these slides to make the point that the timing is pretty good, right? The first FDA-approved siRNA drug, which was based on lipid nanoparticles, was approved only in 2018. So that technology had got to the point of being usable just before we needed it for a different purpose. So the way lipids work in this vaccine production is there's a mixture of lipids that have a few different roles. So we need an ionizable lipid. So you need a lipid that's capable of having a positive charge, because RNA is an acid. It's got a negative charge. So you want our positive charge thing for it to bind to. So you have an ionizable lipid component. And then you have a few others. There's a pegylated lipid. So that's a lipid that places these so-called PEG molecules on the outside of the nanoparticle, and that stops them sticking together. It just helps them to keep as separate nanoparticles. And then we have these biocompatible lipids, the phospholipids and the cholesterol, which help with the structure of the nanoparticle and also help it with its interaction in the body. And so the siRNA gets bound up by these lipids, and they form these little lipid clusters, and they aggregate together to form these larger nanoparticles. And that's how the siRNA gets stabilized. So, of course, people tried that immediately with their mRNA and found out it didn't work. So MC3 is the name of this particular ionizable lipid. There's the positive charge there. That works well for siRNA, but apparently not for mRNA. So what Moderna did is what medicinal chemists do, what pharmaceutical companies do, is they screen lots of different variants of the kind of molecule they're interested in to find the one that does have the right properties. And so they made hundreds of different ionic lipid formulations. And they showed that this one, SM102, works for making lipid nanoparticles to package mRNA and deliver into mice. And so it's a different lipid. It's got the hydrophobic parts over here, and it's got the ionizable part here. So we've now got all the physical parts, right? We've got the protein, the RNA, we've got the lipids. We know what we need. So now we're going to take a quick look at the timeline of how they came together so quickly.
So, 100 days in 2020, what happened to help us develop these vaccines so quickly? So the first thing was, at the end of 2019, the first reports of people with this new undiagnosed pneumonia occurring in Wuhan in China. Roll on to mid-January, the sequence of this new virus was publicly released, and here is the published version of that, although it was shared with scientists. In fact, Australian scientists were some of the first to get that sequence. So it was shared after a few weeks of that first case in the middle of January. So once the viral genome was released, very, very quickly, amazingly, actually, this just blows my mind. A couple of weeks later, there was a paper posted about the structure of the new virus spike protein in that pre-fusion conformation that Charlie talked about. Remember the conformation that would be necessary to stabilize the protein so that your body could have enough epitopes to recognize. So just 2 weeks, because this group had already done, you can see Barney Graham and Jason McClellan, the people that Charlie spoke about, they'd worked on RSV, they'd worked on those other viral spike proteins, they knew exactly the molecular handcuffs and the tricks that needed to be done. So they released this sequence and they said, we've taken the natural SARS-CoV-2 spike protein and we've now just tweaked it with those little tricks. So as soon as this was released, this was actually the critical bit of information that Moderna and BioNTech needed to go and design their mRNA to make this particular protein. So they then, both companies, this is the famous designing over the course of a weekend. This is Melissa Moore, she's the Chief Scientific Officer of Moderna. She has given several talks now describing the process of designing over a weekend. In fact, it was her PhD student who designed a couple of different mRNAs, and one of them went through, and is the one that, if you received the Moderna vaccine, you would have had the one that her PhD student designed in the course of an hour, which is pretty amazing. They used and relied on those bioinformatic tools that I talked about to choose the best elements of the 5' and 3' aspects. And you can see here part of the sequence, and if you see this little funny code in there, that's the pseudouridine. So you can see it's a mixture of C's, G's, A's, and these pseudouridines. So they were able to make their pilot vaccine in mid-February and go into clinical trials from April, which is just amazing. And that's also because the manufacturing setup is the same for any different mRNA sequence. Unlike Charlie explained the really expensive process of making proteins where each one has its individual shape and it's quite expensive, it's actually not that expensive to make mRNA. They were then able to use the lipids that Charlie discussed and put these mRNAs into these lipid nanoparticles and take them forward to the clinical trials. Okay, so those lipids that were first dreamed up by the Canadians were mixed with the RNA and then put into these vials. The next part of the story is actually again going back to the HIV, all the money that was put into HIV research. because part of that was to set up a network of clinical trial sites around America that were designed to test candidate HIV vaccines. So in mid-2020, all those sites were then pivoted to testing COVID vaccines. So all of the protocols and the procedures were in place, and so the clinical trials for the Moderna and BioNTech vaccines were able to go ahead. And it also took a heck of a lot of money, which every government was quite happy to put money into vaccine development because they knew that that was a big part of getting us out of the mess of the pandemic. So roll on to the end of 2020 and we saw results like this of protection against severe disease after 2 doses of, in this case, this is the Pfizer-BioNTech candidate, and you can see that without the vaccine, the incidence of severe disease went up and up and up over time after the first dose, but with the vaccine, we have this very nice protection against severe disease. And both the Pfizer and Moderna vaccines have about 95% effective protection against severe disease. And we're all comfortable and familiar with this story now. Most of us have had this vaccine. So that's really how we were able to, over the course of 1 year, produce these vaccines. And I hope what we've shown to you is that there was actually many, many years of scientific research on proteins, RNAs, and lipids that all came together at the right time. In fact, I've heard Melissa Moore speaking that if it had been SARS-CoV-2 creating COVID-17 or COVID-18 as opposed to COVID-19, then Moderna and BioNTech would not have been at the right place in terms of their technology to create these vaccines. The fact that the virus came at just when it did meant they were ready. But if it had come 1 year earlier, they weren't quite ready. So we would have been using other types of vaccines, or we would have had to wait an extra year. So we were actually incredibly lucky when it comes to this technology. And we in Australia are very excited about this technology now. I was part of a group that actually lobbied the federal government to say that we need the capacity to make these vaccines here in Australia. And they worked with Moderna, and Moderna has announced that it is building now a plant in Victoria to make mRNA vaccines. And our government has put in advance orders over the next 10 years for vaccines, not just for COVID, but also for influenza as well. And this will be the first mRNA vaccine manufacturing facility in the Southern Hemisphere, and it should open up a lot of opportunities for other RNA-based therapeutic products, not just vaccines. So we're all of us in the RNA community in Australia very excited at the prospect that we can develop our own vaccines and therapeutics against other viruses and help in part of this really, really fascinating biotechnology boom. So I'm going to hand back over to Charlie now, and he's going to talk to you about that mystery 4th ingredient that I wanted you to think about throughout the course.
Yeah, so it's an interesting thought, right? As an academic scientist, I study proteins that I'm really interested in, and I find a lot out about them. A huge amount of very careful thought went into a lot of areas to get to the point where we could make a vaccine. But it's more than just clever science that's required. You need something more, and what you actually need is an innovation culture. These developments were done in universities, they were done in medical research institutes, in government research institutes, in completely commercial enterprises, in major pharmaceutical companies, and in new startups. Each of those different types of organisation have a different way of doing science, different level of risk, different level of investment, different ways to work together, to collaborate or to compete. And this is, I think, one of the interesting messages here is that the innovations that came through, particularly through some of these companies like Moderna and BioNTech, were about their approach to innovation and bringing ideas together to make useful medicines for humans. And I think it's an interesting thought for WA, because we are able to do a lot of the things that are required for various biotechnology and medtech applications here. We have experts in biochemistry, molecular biology, cell biology, immunology, cancer, all of these areas. We have amazing synthetic chemists, medicinal chemists, nanotechnologists, all of these kinds of people. We're at a stage now where we've got a biotech sector that's maybe starting to expand. We've got a few more biotech players in WA. And if we can continue helping our government to invest in the biotech sector in these developments, we can start to be really in control of our own future. As medicine becomes more personalised, we will have the tools to deal with our local medical issues more locally through an innovation culture. So what does it mean for WA? It means that we've got something really interesting in the future, I think. What does it mean for you? I don't know, it depends what you want to do with the rest of your life. But I'll leave you with the phrase that if you want to cure 1,000 people, become a doctor. If you want to cure 1 billion people, become a chemist. Thank you.
Very much. Got some time for some questions from the floor. Yeah.
You mentioned at the start that a lot of this stuff came from searching for a HIV vaccine. Why have we not been able to implement it? What's different about HIV compared to COVID?
Yeah.
I don't know.
Neither of us are actually virologists. My understanding as an RNA biologist is it has to do with the complicated life cycle, if you like, of HIV. So for those people who already have HIV, the problem is that it sort of hides and it's integrated into their genome and it hides. And it doesn't actually sort of show itself or produce viral particles. It's in a sort of latency phase. So that's once you have it, that's why it's not sort of possible to be cured of it. But the holy grail would be to have a vaccine, which means you can't be infected in the first place. And I think it comes back to what Charlie was saying about the protein on the outside of the HIV virus being much worse behaved than something like the SARS-CoV-2 spike protein. Having said that, people are developing mRNA vaccines against the HIV spike protein, but they're obviously sort of being developed. They're not proven yet. But it could be that making an mRNA and then having our cells produce the protein, that natural protein that we make might be better behaved than a protein that is made in a lab, which is what we've had to do before. Would you agree with that?
Yeah.
What role does technology play in creating that mRNA? Like, do you have to do that with yourself, or can that be produced through technology?
Yes, so I didn't really go into it, but the RNA in the vaccine that we've had is essentially just made in a test tube, or it's a reaction vessel, where you put in the DNA, the enzyme, and the building blocks. And the enzyme creates all of these RNA molecules. And then they have to be purified. You have to make sure that they're not fragmented in any way. So there's quite a bit of purification and analytics. Then it gets mixed with the lipid nanoparticles. So there's a lot of technology that goes into creating that RNA, even though fundamentally what's happening in that reaction vessel is really similar to what would happen in our cells to make RNA. But we're copying what happens in our cells.
That's kind of what biotechnology is. It's taking those things that biology uses and extracting them and using them to our advantage as technology. And making the RNA is exactly one of those biotechnology applications.
Question about?
Can you comment on the function of the actual COVID spike protein? Like you mentioned that actually do? Like, does it help attach to our body cells? Does it create a docking station and then a funnel where it injects its own genome? Is that what the function of that protein is?
So the spike protein binds to a receptor on certain cells of our body called the ACE2 receptor. And once it's bound to the receptor, it becomes, the whole particle gets internalized. And so it's bringing the viral genome into the cell. And then all those other proteins that Charlie talked about, the structured and the non-structured proteins, they get to work and they make more copies of the viral genome and produce more viral particles and then secrete viral particles. So the spike protein is absolutely needed to get into cells. If we could remove the spike protein from SARS-CoV-2, it would become non-infectious.
And that's why it's kind of the weak point, the Achilles heel of the virus in terms of vaccine production, because it's designed to interact with us, so therefore it has to be available and recognisable, and that's why it's a vaccine, a useful vaccine candidate.
So at this stage, I mean, you're creating, or you're looking at recognisable parts of that protein. What about, is there some way of kind of jamming up the function of that protein?
Well, that would be, I mean, that would be how some antiviral drugs work. I don't know specifically about anything applied to the coronaviruses, but there are, the drugs, anti-flu drugs, for example, work in exactly the same kind of way. They interfere with the ability of the body to take up the virus, so they interfere with that interaction and stop that being possible. So that's more of a drug type approach.
Yeah. Is there anything different about, you mentioned that the specificity of the RNA nucleotides, we get like a methyl group attached to the N1, but we were doing them in mouse models, so is there any different, like, do they still have that same methyl substitution on the nucleotide, or do they have a different one with that effect using?
Yeah, so actually the N1 methyl pseudouridine was identified in a completely different microorganism, I believe.
Archaea.
Archaea, yeah. So we don't naturally make that. Nevertheless, it's interesting, isn't it, that it will fool our cells into thinking it looks like a human mRNA, even though that particular modification has been observed in archaeal mRNA. So I think that's why I said, it's such a big new field of understanding what all those RNA modifications are doing and why we have them, because we still don't actually quite understand. We thought for so many years that it was just A, C, G, and U. So, yes, it's a really fascinating area. But in mice or humans, if you put in pseudouridine or N1 methyl pseudouridine, it will have the same effect of fooling those cells into thinking it's not synthetic.
At that level, we're not very different from mice. We're kind of the same. I mean, when it comes down to amino acids, we're the same as bacteria and every other living being. But modified nucleotides may be a bit higher up.
So over the last 2 years, the virus has mutated a lot. Are we still using identical messenger RNA vaccines as the original ones, and is that plan changing?
So we are still using identical ones. However, both Moderna and BioNTech have Omicron-specific vaccines that are currently in clinical trial. And it may be the plan to have boosters which are Omicron-specific. At that point, it might have evolved further. And indeed, that's what happens with flu vaccines. So every year in the southern hemisphere, we look at what's circulating in the northern hemisphere, and we design usually 4 different variants based on that and create our flu vaccine cocktail. So we'll probably end up at a point with COVID vaccines where We will have a cocktail of slightly different sequences.
Yeah, I think the first attempt at an Omicron-specific one didn't show any different efficacy to the original one, so that's why it didn't get rolled out. But obviously they're working hard to find new ones that do.
So in Australia, we're actually developing our own homegrown mRNA vaccine that's a little bit different. It just contains the very tip of the spike protein as opposed to the whole spike protein. And that's in phase 1 clinical trial at the moment. Yeah.
Okay. Yeah, one last one.
So clearly Moderna and Pfizer made a huge amount of money, far
Yes.
more than the amount of money they say invested up front. How much money did the PhDs do?
That's a very good question. You would hope that he would have got something nice.
I
So
know someone who was a PhD student who designed one of the drugs that became one of the HIV drugs, and he was unable to have his thesis made available for about 8 years after he finished his PhD, but he was given a very nice job at the company that he collaborated with during his PhD. So I imagine this PhD student probably got some reward.
Okay, there's some light refreshments outside. Please join me in thanking Charlie and Arthur.